I'm sure.
Okay, well, it looks like, there are people filing in, but I think we can go ahead and get started then. So again, thank you everyone for joining us today. We are going to, start off with just some updates, and then I'll hand it over to Noel Burtt, who is our featured speaker. And just a reminder to everyone, if you have any questions, please feel free to use either the chat or Q&A. We'll be monitoring them throughout. And also, happy Halloween. So I'll just start off with just some reminders and some updates. If you were unable to view any of our other webinars from the previous sessions, this is our YouTube. This QR code will take you straight to the page. We have every single one of our webinars available there that you can play through back. And if you have any feedback, questions, or would like to be added to our listserv, please let me know. I can add you. Just email us help@cfdeknowledge.org. And I just want to remind everyone about the incredible CFDE podcast that we have, which is hosted by Alyssa Dillman. And this is, not just a podcast, it's a community for passionate individuals who are curious about omics research, who might have some questions about biological puzzles and addressing health concerns. And so, one thing that I just want to note for this is there are now 10 episodes that are out, and I know another is on the way. So be on the lookout for more episodes from this podcast. I just want to take this moment to reflect on the American Society of Human Genetics this past month in Boston, Massachusetts. We had a great showing for the Common Fund Data ecosystem across various events that we held throughout the week. We had a brand new working networking session that we had to kick off ASHG this year. And thank you to everyone who participated. We had a great few hours of sharing science, meeting new individuals, and just getting really excited about all the ecosystem has to offer, both inside and without it. And we also had a fantastic CFDE booth. This booth had different spotlight hours with different programs throughout the week. So thank you to everyone who stopped by, said hi, and learned a little bit more from our various programs. We also had a very knowledgeable ancillary session where we walked through some of the scientific highlights that are throughout our ecosystem and featured the Knowledge Center, Training Center, and SMOT. And so we're very excited to have all of the research there be available to everyone within and outside the ecosystem as well. So again, big thank you to every single person who participated. We are going to be obviously attending more conferences from here on out. So be on the lookout for other places that you can talk to CFDE in person. And so with that being said, again, happy Halloween. I'm going to hand this over to our featured speaker, Noel Burtt, who is going to be presenting on one of our enhanced workflows and basically giving you an insight on how you could transform research questions into mechanistic leads. So with that, I will hand it over to Noel.
Thank you, Mackenzie. All right, I'm going to share my screen and then I will give it a shot. So let me know when you see everything. There we are. Can you guys see everything okay?
Yes, that looks
All
great.
right, fabulous. All right, I'm going to move this little Zoom bar always is a pain in the butt. All right, before we actually dive into the actual feature I want to demo today, I want to, because some of you don't join our webinars all the time, some of you are new, some of you are in the ecosystem, so this will be a little redundant for you who actually are collaborators in the Common Fund Data ecosystem. But I wanted to sort of just zoom out and just give a level set to what the Common Fund Data ecosystem is, because I think it's really critical in the context of what I'm going to share with you today. And also to begin, this is actually really exciting for us. It's like literally brand new. We actually unveiled it this Monday as we all went to our actual all hands meeting for CFD and shared it with our CFD colleagues. So it's brand new and it may break, but it's more that it's a workflow that might take some time as you're working with it. So I'll show you as you go in. So we're really hoping to get your feedback on it, to be honest, how it's working. Is it useful to you? And what you really would like to do with it if it doesn't do that yet. And that's kind of like, so have that in your head as we go through this conversation. But with that sort of the common fund data ecosystem, just to remind you of it, is an ecosystem within a broader ecosystem of NIH resources and other, data science platforms within our universe across genetics and genomics. And I'm sure you in your research use these resources and tools. I'm going to give you a little bit of an anchor to that, because I think what we're going to talk about later actually has implications both for inside the ecosystem and ideally externally, because I think that's really what NIH's goal and many of our goals are as we do these research projects. We use a lot of resources. We want to minimize the places we go to and create, better, interactions and interconnections between these resources to create more meaningful hypotheses around biology and mechanisms of disease, right? So today we're going to really focus on enhanced workflows for discovery and knowledge, because that's really what we focus on within the Knowledge Center. And the way the Knowledge Center thinks is four types for knowledge. One is foundational, which is literally, the basics, how a study was conducted, why it was conducted, the data it's generated, and so information. So our job at the Knowledge Center is to pull that, all that knowledge from all of these resources that are working with us into one place and make them accessible for you. And they're curated with our colleagues at all these data resources. The second is secondary knowledge. These are things that are derived analyses from either one CFD program or multiple ones. And these are either analyses that the data resources centers have done themselves and may not have them made accessibly publicly, or we do in partnership with them because they're like, hey, we've never done this analysis. We want to connect it to human tissue or human disease. And we have a sort of agnostic analysis within our data resource. So we work with them to do that and make those accessible. And the last is really the fun, which is tertiary. These are assertions that are conducted across multiple common fund data ecosystem programs or with external resources to derive knowledge and results. So just that's the framing for all of the results you're going to see on the Knowledge Center. And the Knowledge Center really is about summary representations and basically creating a first glimpse of the common fund data ecosystem, because the resources within all have their own specific, niches within the environment where they house the primary data, or they create ways for you to view the data in their own context, right? Very different than what we do across the entire ecosystem. So with that, just sort of thinking about how you might have stumbled across the CFD through your time. You might have seen it through the NIH website. You may have never heard of it. You may use a CFD resource and notice that, oh, what's this member of CFD? So this is on GlyGen. They do a great job of sort of saying, hey, we are our own consortium and we cover glycobiology and glycoproteins, and there's an entire resource right here, but there's also this other resource. So that's another way you might have learned about the common fund data ecosystem. Another one might just be by mouth. When GTEx is talking, they may be like, hey, we're part of the common fund, but it isn't always clear. And I think as these resources evolve, they are 10-year projects and they are not going to be around forever, but the data and the tools and the discoveries within them should be around forever to be used by our community. And that's what we're trying to do within the Knowledge Center to make this accessible. So another reason to create an ecosystem is because of what I was just mentioning, but they're obviously quite diverse and they all have their own communities and they may not make sense to come together. Like, why would you think that? But there's some themes and threads. And the themes and threads most of them are data aggregators. They either bring data from the public or from the actual data resource itself with the consortium and integrate them. They create reference maps, like HubMap creates a reference map for all human tissue for spatial and single cell data. Or they create molecular signatures like links, where they perturb a lot of genes in a cell line and read out those signatures and make those accessible, right? So these are all themes and threads. But a commonality core value of all of these resources is they're all fair. They all have their own communities and they all really think about making those data accessible. And they also really are quite good about engaging with their users. But you can imagine across the ecosystem, we take these same principles and we bring that to another level. So that's why the ecosystem, that's why we enable a resource for you to query across all the common fund data sets and resources to sustain those tools and create new findings from them. And also we really want to make sure we train users in all the common fund data. That's one of the pillars of the common fund data ecosystem. So what's our niche? Our niche is really about providing knowledge of the entire ecosystem. And so I'm going to talk about how we do that. So one, you may have used the resource. Just if you go to cfdknowledge.org, that's where you want to begin. And all of the rest of the conversation today is going to orbit around this entry point. This is not the only entry point to the ecosystem. That's what's so great about it, right? You can enter through a DCC, a data coordinating center, and get here. You can enter through the data resource center, for example, and get here. And I'll show you some of that later. But we're going to start for the purpose of today in the Knowledge Center. So there's a landing page. You can type in a query, whatever you want, but let's talk about that first. But maybe you're not familiar with the ecosystem at large. So the first thing we want to do is provide sort of just a snapshot that's interactive of the entire ecosystem. So if you hover over this, you can get to any of the DCCs and you can learn kind of like what are the entities, what are the omics data, what are the specialties of those resources and what they are. And then you can also learn that to support this entire resource, there are a set of these 5 centers. A coordination center keeps us all in line. Training center meant to train across all of these resources. The Knowledge Center, us. A cloud infrastructure, which you'll hear about a little bit later, that allows you to take data from the ecosystem, bring it into a workspace, and do compute on it and analyze it. And that's a newer resource, which is really great because it connects all different user types and allows people to do their own analysis on CF data. And then there's the Data Resource Center, which allows you to get the raw data and the metadata, and also has a host of tools that have different ways that you can interact with Common Fund data. So those are the centers. So together, that's the ecosystem, right? Okay, so now connecting features. They're connecting features of the ecosystem, and this is really what their Knowledge Center is kind of based on. The idea that if you look across the ecosystem, you may think of GTEx when you think of transcriptomics, right? It's the one place you would go to look up the expression of your favorite gene. But it just so happens that there's a host of other resources within the Common Fund that have expression data. Therefore, as a user, you might really want to have the benefit of saying, well, if GTEx has a pipeline for doing differential expression and has like kind of the best like foundational resource for gene expression, wouldn't it be great if other gene expression data from the Common Fund could go in one place and I could do differential expression, for example, analysis on all Common Fund data? Because some of the Common Fund data has disease-specific data types. Some of them, like MotorPack, for example, it has exercise-induced gene expression. And those things could be analyzed together and sort of gives you a sense of, gene expression for your favorite gene in the context of both disease agnostic resources and also disease specific. So it gives you a flavor of all the different types of things. So it serves as a tool or a reference, right? So that's an example for gene expression where the ecosystem brings all of its data together and allows you to one centralized analysis. So what we do in addition to that is we think across CFD and we think, what are the types of secondary knowledge? These are things that are derived from an analysis of either one Common Fund program or multiple in some cases, and external resources sometimes that are analyses that aren't commonly done by the data coordinating centers because their job is really focused on generating the data and making it accessible. So we have a carousel of these, and we do these across all the DCCs, and we're looking for more to do because these are kind of the ways to create connections within the data coordinating centers. So one example, Julie, yeah?
Yeah, I was just going to say we got a question from one of our participants, Natalie.
Sure. Hey, Natalie.
So Natalie said, I'd love to understand if there are ways to query which CFDE data sets are open access and controlled access.
That's a really good question, Natalie. There are, I think, not on the Knowledge Center, but I think what we've got in the Knowledge Center, I'll answer your question in 2 ways. One is if you go to each of these DCCs has a page that we've created, which is like our primary and foundational knowledge. And that, I believe, Julie, you might be able to answer this better than I. We summarize the data within all the resources. Let me give you ways to get to the data. I don't know if we explicitly say on those pages whether it's controlled access or open access, but I think what we try to do is bring you to them. That's one way. And I think the DRC also has a sense of, for the data that is open access through the C2M2 model, which is the common data model that we've had across the CFD, that data is all considered open access. And then for anything that is protected, in many cases right now, I believe that is something you got to go to the data resource centers to find out whether it's protected or controlled. But it's a really good idea for what we're thinking about in our next release. So you're showing our future, which is what we want to make it so is if a user has no idea, for example, like, hey, they want to like look at kids first data, we actually want to allow you through like an enhanced workflow to say, I have a question about kids first data. Is it open access? And we would actually answer that back through, a RAG-based text embeddings that we're going to incorporate within this front end snapshot that would give you a glimpse of what's controlled access and what's not. So that's coming. And I think that's a great thing for us to think about, Julie, to make better. Did you want to comment any more about that, Julie?
Yeah, no, I agree with what you just said. In terms of the foundational knowledge pages that Noel just mentioned that we have for each CFDE program, it will specify in some cases where it's very clear that, there are clinical data or something with protected health information that a user would need to apply. But I think your idea is actually fantastic if we had a way of tagging, this DCC has controlled only, open only, or some hybrid of the two, at least so that users would know what they would have to apply for versus what they could get readily. I think that's a great idea.
Yeah, it really is. We'll call it Natalie's idea when we have it up there. Thank you, Natalie. All right, cool. Hopefully that answered your question, Natalie. So then, in terms of representation of secondary analysis, a couple examples of this are for IDG. They've done this incredibly good job of saying, like, for a given target, if you want to see if it's druggable, you immediately have a classification of that. So that's knowledge that they've derived themselves that we make accessible in the Knowledge Center. So if you type in your favorite gene, you're going to get that card right away. But then, for example, there's other information like for glycan. Is my gene glycosylated and how is it? That's another representation. This is directly from the resource, but this is stuff that, like, immediately when you go to that specific resource, there's a lot of other things that you may not want there. So if you're a geneticist, you may not understand, all the other stuff in that resource. That's why here we sort of bring it down to a context-based view based on the gene and the fact that you might want to know everything about a gene that the Common Fund has to tell you. Sort of like those glimpses. That's just an example there. And allows you to drill down to the specific data, and that's directly from glycan. It's an example. Another example is HubMap. HubMap is, I don't know if you ever used it, it's absolutely beautiful. It's one of the most beautifully represented of data with interactive visualizations and like really intelligent ways to drill down to like what they call their functional tissue units. You can see all the parts of their tissue units and where they're derived, and it's all anchored to this like human reference atlas, right? So that's about reference. So what we do is we allow you to get kind of a glimpse, right? So we've created integrated maps for all of their tissues and allow you, if you type in your favorite tissue on the Knowledge Center, you will immediately get the glimpse of all the HubMap data curated with gene expression data. So you can do a quick lookup of your favorite gene, for example, on the Knowledge Center. So again, it's all anchoring CF data into like a context, right? That's around a scientific question to give you a glimpse, to sort of like give you the first level. And this then requires you, if you're so interested, to enter the ecosystem, to go deeper, to get at that data, or to ask more questions of that data, and so on and so forth. For example, another one is Kids First. Kids First is an example of protected data. These are children with pediatric disorders. They're very small cohorts. Therefore, summary level representations are not available anywhere else. We've worked with Kids First to make those summary level representations available. And so we worked with them to take their data, their protected data, run an analysis for it with them, variants, to determine, variants associated with their various phenotypes. So you could do a lookup within our resource of your favorite gene to see what Kids First says about it for those phenotypes. What's cool is this allows you to see things that, like, for example, are not in Nomad, that are in the literature. So this is validated results, but it's not readily accessible on Kids First, but enables you to get a quick glimpse of what Kids First could offer you, right? But also it allows you to integrate this knowledge with your knowledge about your favorite gene or condition that you're studying. So what's great about this is that this is something Kids First could use in their work, because it could be on Kids First site, and you could look there and say, oh man, I want to see this. Now I want to get a request access, as you said, Natalie, for the underlying data for that. And that would take you through that process so that you can get access to that data. That gives you another example of a secondary analysis. Another secondary analysis is a really cool one that we've been doing with 4DN to understand the relationship between human phenotypes and what gene they're connected by non-coding variation and, DNA and chromatin structure. So for this, we've applied a novel analysis to 4DN data we call Falcon, which basically allows you to determine which human genes are connected to a phenotype and which variants drive that connection, because this is across the genome. So all the non-coding variants have these analyses conducted on them. So this is another way for you to say, I have a favorite human phenotype. I want to know what 4DN has said about it, how it's connected to a gene or phenotype of interest. So this is also available in the Knowledge Center as well. But again, if you want to get down to the data underlying this, we provide a way to step through. And for example, 4DN data is, Natalie, to your question, open access from my understanding. Another one is MotorPack. MotorPack is a really cool resource about exercise, and they do studies on humans and rats. And they have a gene-centric view. And their gene-centric views allows you to look up just MotorPack data in the context of your favorite gene. But what if you were on MotorPack and you're like, I'm studying FTO, I want to know what else is known about it within the Common Fund and beyond. If you click FTO, it used to take you just to the, the gene page, the NCBI gene page, but now you're going to go to the Knowledge Center where you'll see the hub map connection. You'll see the Kids First connection. You'll see everything for that gene in the context of the Common Fund data ecosystem. So this is a nice way of just interconnecting across an ecosystem. So with that was just the blitz of information about the primary, secondary knowledge that we represent in the Knowledge Center. But that's a building block for these tertiary workflows. These are really the fun things. These are the things that require us to think outside the ecosystem, because that's really what's useful. Many of you are not working within the Common Fund data ecosystem. You might know of it, but you have your own analysis that you're doing. You have your own set of experiments. You work in a mouse model, you work on, programmed cell death, you work on a particular processes or a biological context, or you work on just a disease. So we want to create analyses that layer data from the Common Fund with other resources to supply new hypotheses and ideally mechanistic insights, but also to do it in a holistic way so that it's also a substrate for engagement of other resources, because as you know, there's a lot of other resources out there that are doing really great things as well, right? So we have three resources we call our early workflows, which is one is the mouse phenotypes to human phenotypes. Another one's the gene set analysis, and the other one's differential expression, which I showed you earlier. But this is like the entry point to something else I want to talk a little bit more specifically about. So this is about what we call enhanced discovery. And so this is going to be the context of the rest of the conversation today. So we do the enhanced discovery, to me, based on a commodity within the ecosystem. It's not the only thing within the ecosystem, and it's not the only thing we study, but it is incredibly ubiquitous within our environment. That's genes. So it is the fact that many Common Fund programs have in common the fact that they're informing on biological mechanisms through genes, producing gene sets or doing analyses on genes and producing results, right? Genes are simple, concrete, and easy to generate because genes are something we know about. We can study them. We know a lot about them in the literature, and we know we have sequences on all of them, right? Gene lists are easy to generate because they're small. They're not like big BAMs sending around, BAMs or bed files of all these big data sets. They are summary representations of an experiment or a perturbation or an activity. We have an infrastructure, which I'm going to share with you in a second, to capture and analyze them. So what's useful is these gene sets can infer biological mechanisms, and they can also help capture those mechanisms by where they were derived from. And they also can ideally be relevant to human health. So they're a commodity and they're useful that way, right? So we are very lucky to have collaborators at the DRC who are kind of leaders in generating gene sets across the Common Fund data ecosystem. And they've generated hundreds and thousands of these, which is awesome because almost all of the Common Fund data ecosystem resources produce gene sets as a result of their experiments. So Avi Maya and his team have captured all those and created gene sets for over 277,000, actually I think more now, gene sets. And we collaborate with him to bring those in to the Knowledge Center to combine with human phenotype. So that's the substrate. So we have all these gene sets that are derived. And what we do is we link them to human phenotype. And we do that by another set of resources, which brings us external to the CFDE. So genome-wide association studies, right? So what's great about those is they're genome-wide. They assess genes all across the genome, but they also have been done in thousands and thousands of phenotypes. And with our other hat on, in the human genetics world, we run a series of knowledge portals for complex disease and rare disease resources that we bring data together for GWAS data across what we call the Association to Function Knowledge Portal, where we have about 1,300 curated phenotypes for human phenotypes, mostly common disease. Then we work with the GWAS catalog to bring in all of their other diseases that are not in the A2F. So that represents a set of genome-wide associations. And then for rare, we work with OrphanNet to bring in all of their association results. So these are summary representations of common rare disease. So that's another substrate. So these are all brought together to create a substrate for interaction. And so we've worked over time to add new DCCs, just to mention that we're very lucky that we're generating new gene sets across the ecosystem. So within the resource, we have a way for you to navigate through these. So if you go to on the top header of the landing page, you'll see analyses. If you drop down, it's like a gene set browser. This allows you to browse and curate and play with these in the context of the Knowledge Center. But today we want to talk about something to like bring it to an easier space. This is very detailed, and if you know what you're looking for, you want to look at gene sets from Spark, for example, you want to look at gene sets from HubMap, or you have a phenotype you want to go after, this is probably where you want to go. But if you really want to drill down deeper, I think what I'm going to show you might be a little bit useful, because the activation energy is huge, right? Like, so what I just showed you is a lot, but also there's a lot of other resources out there that can do this really well for you. And I imagine as an investigator, particularly someone in the experimental biology world, it's a lot to go through all these resources, and it takes a lot to do research. It's exhausting. So the gap and the gap which we hope to fill and the innovation is sort of around the idea that the cost benefit analysis, all right, that you need multiple approaches when you're doing research. You might want to download and integrate data, right? If you're doing a differential expression analysis, the cost is high, but the benefit's super high too, right? So that's what's great. So that's one mechanism by which you'd actually do research to, follow through to get some initial hypotheses that you then might follow up. That's a lot of work. Cloud-based workspaces are a little bit less costly and slightly less benefit because, you have to go in that environment. A workbench like the CFDE kind of brings it all together, gives you some tools. That's great. You still have to figure out how to play with the data. You still got to know what to do. You still have to be pretty computationally savvy. But then there's portals that are pre-computed, right? So they tend to be more on lower benefit, but really cost. They give you a quick glimpse. That's really where we sit. But a user kind of has to span through all of these over the course of a research project, because you really got to get the activation energy to develop a proof of concept. And then after that, it's kind of a little easier. So, when you do lookups, you might want to use a portal for a preliminary first pass. That gives you like an interest, like, I might pursue this question. Beyond that, you might, look up again, go deeper into it. This gives you the proof of concept. I see this result. I see this association to a gene set. Okay, now I'm in. Now I want to go and analyze the data. That's when the benefit goes up to really learn it. But again, the cost goes down, right? And then finally, full analysis. That's the full enchilada where you want to get to, and makes life a little easier. And that's the progress of the project. What we want to do is move this space right here for that lookup result and make it a little bit more beneficial, deeper cost, and then lower that barrier for activation so that you can get to doing a full analysis and full project faster. That's one mechanism, that way which we care to do this. The other one is to do that in the context of very different users, right? So what's great about the data resource centers, all those 18 of them that I was talking about, they understand their user way better than we do, right? But we want to bring all that data together. So we have to think about how to create an interface by which we can easily allow the user to share their contacts with us. And therefore, the results that we retrieve are data-driven, right, based on the CFD data, but also in the framework of the context of that particular user. So that's what we're hoping to do. So two ways to do this. And it's all based on one foundation. So we do what we call create a genetically calibrated knowledge matrix with two main components: human genes, all of them across the entire genome, well, 20,000, and then human gene sets derived from the Common Fund data ecosystem, of which there are about 200,000 right now. And so these are things that I was mentioning earlier that are contained, derived. We know them. We can document the province. We know how they're made. We know who created them. So these are two very strong substrates. There's 7,000 phenotypes here across all genes in the human genome, and these gene sets that span programs. So these two basically raw materials go into this analysis, which combines these to create connections between the phenotype, the human genetics, and the gene sets to help you derive hypotheses. So there's a lot to unpack, but let's go through it a little bit together. So it's based on this model. It's a statistically principled framework. It's called Pigeon. I'm sure you've heard us mention it and beyond, but the idea is it uses indirect and direct evidence to connect human phenotype to a gene, right? That's very simple. It's much more complicated mathematically, but it's a very simple concept. And we do that because we have these three prongs, which is phenotypes, all the genes, and all these gene sets. And we pull them together to create a statistical probability of those results. And that creates essentially a knowledge graph that is anchored and structured based on statistical inference. So this is kind of like a proof of how it works. If you look at all these other methods, this is from a study of basically druggability of genes based on human genetics. So this is the idea that pigeon basically moves the needle, if you see at the top, for all of these data sets and makes it has a slightly higher relative success for these targets. So it basically is a better model than our currently models that are out there. So that's why we know this works, and it's statistically rigorous, right? And the idea is that it links predicted drug targets and estimates the success. So this is essentially at the end, it's like a matrix of phenotype, gene set, gene triplets as an entry point. So this matrix is essentially a knowledge graph. But how do you navigate through that? That's a lot, right? So a lot of you have used knowledge graphs. They're complicated if you don't understand them, and they all have something that brings them together. This is about genetic data. But the uses of this are pretty awesome, right? So they can be integrated instantly with knowledge graphs, but they also can serve for like a lower complexity interaction as like a backbone or a map, right? So this is a complicated interface, and this is like, wow, there's like traits and genes and gene sets, and you're like, how do they work together? Well, this enters an enhanced user interface or workflow so you can navigate through this and start to make sense of it. So for that, we've developed what we call Reveal, which is a reasoning engine for validating evidence across the landscape. And this is across both the landscape of CFD and beyond. And it's based on a semantic search, basically in probabilistic model that I just mentioned earlier, called Pigeon, and an LLM-assisted reasoning to reveal, ideally, new connections between CFD programs and human phenotype. So how it works, again, this is based on a lot of work from our team, but again, you describe your research context, it parses these search terms for you, basically acknowledge like, hey, actually, let me just take the next in action. This is a little bit easier. So if you go to the landing page for CFD, and you scroll about halfway down, CFD, Reveal and Design are right there. I'll talk about design in a minute. But if you click on it, here's where you'll land. You'll land on a simple search box, which says, just ask me a question. And here's some examples that you could run. Like, I'm looking to find mechanisms that underlie body mass index in humans and tissues that are involved. Or you can say, I study circadian rhythms, and I'm interested in common fund programs associated with the liver. That's another example of one. Or anything you want, you can type in. Then the next step is we use semantic-based search, and we basically use like an AI agent to say, here's what I understand your query to mean, right? We say, like, we sift through it to get to ontologies to match your query to a set of phenotypes and keywords. And then we say, is this what you want? And this is the context. And then it retrieves associations from those 7,000 associations across common and rare disease and the gene sets, and says, based on the probability that is defined by the Pigeon method, it says, these are the ones that I think are the most significant associations related to your query, and presents those to you. And then it actually goes out and creates an LLM search that then creates hypotheses based on the groupings that you see. So that's it at like first step, but let's go through it in a little more detail. So here gives you an example of one of the ones, like, I'm looking to find mechanisms that underlie body mass index in humans and tissues involved. All right, so it says, I heard you say you want body mass index, humans, tissues, mechanisms. And the context is that you investigate mechanisms underlying BMI in humans, and you're interested in that regulation. So then it goes and gets the 300 top associations for that and posts those for you. And this is what it looks like, for example. So you'll get a table of the phenotype, the similarity score, basically how similar is this? And basically it gives you a confidence score. And then an association. This is really what you want to look at. Moderate is just the result that's on the top here, but strong is really probably what you're most interested in. But you can tailor these results. So you will then see the source and then the signature by which that gene sort was created. These are converted from the DCCs themselves that for us to figure out what the source is. And so this is all text-based curation. And then you have the gene sets that underlie this association. So it's a lot. But what you can do here is start to basically play around with it based on what you see. You can pick and choose the phenotypes that have been semantically associated with your BMI query or whatever you put, and you can click and pick and basically filter those based on things you're like, I'm not interested in agent menarche particularly. I'm interested in all the others. You can sort and filter those. But what you may want to do is just like drop out the things that are moderate or low, because those are like not necessarily statistically as rigorously associated. So here I'm picking strong, for example. Then you can also see through the different sources, right? So some of these are just GTEx, for example. Some of these are crossings where we've actually taken gene sets from one resource and another and actually crossed them to see the overlaps. Those are even more gene sets that are at your disposal within this analysis. And again, you can get at all those sources through, clicking even deeper. So then once you've sort of filtered down based on, either you can run all 300 and get an LMM retrieval query, or you can say, okay, I just want to kind of filter it by the strong ones. And then what happens next is if you hit reanalyze at the bottom, you'll get a mechanistic hypothesis. Now, remember, this is a hypothesis. This is based on just the data that you've contained within the Common Fund and within the data we've curated for GWAS associations. So again, it's not spurious data. It's data we've curated and that are analyzed within the Common Fund. But the hypotheses are generated by an LMM that's been trained on those associations, okay? So then you're going to get a couple hypotheses. I think there were 4 on this page, but I didn't put them down. So this is the top one. And what it does for each of these hypotheses, it gives you the programs that are contributing to it, the strongest linking genes, the top ones, not all of them, and then the relevant associations. So you can see its work essentially. And you can say, and then you can view the genes, same sort of thing happens here, but it gives you a little hypothesis. And like, you can say like, oh, I'm interested in that. I want to drill down deeper. So drilling down deeper means a couple things. One, you can just, be like, okay, great, this is interesting, move on. But one is you want to explore. You're like, okay, I actually think this is something I want to go after. And I want to understand like, oh, these 3 genes, I've seen these before. And I think this is an interesting thing. It's involved in lipid storage. Okay. So you can have 2 options here. You can either go and run what we call a validation experiment, which is really exciting. I'll talk to you about that. But then you really want to explore. You're like, okay, I don't know if I believe this. I want to know where the data came from. I want to understand this association a little deeper. So then you can go here, click explore, and it'll take you to our explore intermediate step, which then takes those gene sets, takes your query, takes your hypothesis and says, all right, time to start exploring. And what it will do is review your gene sets, bring a hypothesis with you, and basically allow you to explore several options, of which these are going to expand over time. But the first one is, hey man, I want to get right to the data. 2, I want to understand the scores. And 3, I might want to use all those awesome CFD tools that you've talked to me about to play with some of this data and run in some analysis related to just this data that I've sort of sectioned off to summarize and work with. So 3 things we've done so far, just to give you an example, are, and they are sort of teasers of the future, is 1, like if you pick GTEx and you're like, okay, this came from GTEx, I really want to know where this came from, what this means, why it happened, and so I can believe it or not. You go here, if you click this, you go directly to those genes in GTEx. So then it brings you right through to that data resource center. Another one is, okay, if you MotorPack. So MotorPack, this is even cooler. So we've worked with MotorPack to say, all right, now if you want to just look at those genes in MotorPack and run all of their specific analyses that they do better than anybody else, like gene coverage, interactive tables, trajectory plots, heat maps for just those genes that you've selected based on that mechanism hypothesis, you can go to that and it will gather that, pull it into a workspace and allow you to play with it. The coolest part of this is you can do this in MotorPack, or eventually our lovely collaborators at the Cloud Infrastructure Working Group will have all the data there and you can play with it there for free. So that's how we're growing to really make this ecosystem a place that you can not necessarily leave to do all sorts of cool analyses and discovery. This gives an example of what this looks like. And this is, again, for the data, the genes that I have just said, I think they're part of this one hypothesis about like liver storage. I'm sorry, a lipid storage in the liver, brings it into MotorPack. And then you are just then now in a playbook, if you will, to say like, for these genes, I want to look at all the visualizations that MotorPack has for these genes. So I understand how MotorPack derived this gene set and what I can learn about it. So that's a really cool thing. The other one is back here, which is the playbook. So this brings into all the tools that the DRC has created. Everything you can imagine. There's a lot of them that are involved with this. So it allows you to say like, these are the ones we just give an example of these different playbooks that you can generate through the DRC. But what will happen is if you click that, it'll bring you to the playbooks associated with this set of genes and what you can do with it. So again, it's these tailored based context specific next steps that are derived from a hypothesis that you've made based on gene sets that are in the common fund to human phenotypes. And it's genetically backed by literally like all the genes in the human genome associated with phenotypes. So there's like a statistical probability at this backbone that then is bringing you to these next steps. That's really the key. It's like a hypothesis generator that allows you then to decide whether you think it's accurate or not, but it really does have some rigor to it. So that's where that Explorer takes you. So if you really believe all this, the next level down is design. And what I mean by this is sort of even newer. And this is something that we're really hoping to work with experimentalists on because we're not ourselves. And this is, okay, now I have some genes and I, for example, study energy balance. I have ways to measure indirect telemetry in mice. I'm set up to do it. If you give me a gene, or if I have a gene of interest, I can run them in the mice and basically read out based on different parameters what happens to these mice. So if you have a hypothesis, I can actually execute on it. But bringing, biologists to this may be difficult. So we're really hoping to work with like computational folks and bring the experimental biologists to this. But one way to do it is with this resource called Design, which basically takes the work that you got in Reveal and Explore and all those gene sets that you've curated through a mechanistic hypothesis that you worked with an enhanced workflow to generate, and then provides a way to experimentally plan for you. It could potentially give you plausible assays, relevant cell or tissue types, and also potentially candidate biomarkers. Again, this is LMM assisted, again, but it's in the confines of the data and the connections we've made through the genetically calibrated knowledge graph that we've made. So this is a newer version, a new extension of Reveal. And again, use a lot of testing, but we're hoping this will allow more experimentalists to use the common fund data to bring their research to results that are in the common fund that they might be able to help validate and create discoveries. So that's kind of how it works, a little summary of that. And essentially, that's kind of like a lot that I put out there. How am I on time? Hopefully I've stopped with enough questions. But basically the tagline is, please give it a shot and tell us what you think. Try a question. It may take a while to return. It's not super fast. I think, I don't know if our engineer's on the call, but typically like the first one takes like a minute-ish, and then the mechanistic insights probably take another minute. So be patient. It tells you what's going on. So it'll let you know if it times out. But if you use it and it's helpful, give us that feedback. If you're like, wow, I really liked what I saw, I would love you to add this. Or another thing is like, this is not doing what I want, or I expected this. Like I had great feedback from a person in Spark. We have Spark gene sets in here, but Spark really isn't about genes. It's about, basically the signals that happen in the body across, from the brain to the hand, and that like electrophysiology mapping. And that's not necessarily easy translatable to genes. So what we're hoping over time is, how do we expand this resource that's like anchored to genes to things like physiological, processes? We may be able to understand how genes are involved in that right now, but learning about what Spark has actually found, again, we're just sort of dipping our toe into that water. So there's a lot to be done, but again, using it is key, and telling us what you think about it is also even more key. So today's webinar is really about just sort of sharing with you what we've got and where we're headed. And again, we hope you take a look at it. A couple things to mention as well before I break. Actually, Mackenzie mentioned this earlier, but so the future in the next year is, there's three things that might be of interest to you all listening to this, that we have been very blessed to work within the Common Fund ecosystem to enable events that will really bring together NIH resources and folks outside the ecosystem to experimentally validate the results. So the first one is in sometime in 2026, we hope, once the NIH opens, hopefully that'll happen soon, is one of the things we really see CFD as is a catalyst really for NIH's mission to reduce redundancy, to enhance data utility, and ideally to accelerate scientific discovery across a lot of these resources that the NIH has, right? So CFD is an example of very large resources within NIH coming together to create a meta resource, if you will. I think that's a great model. And so we're hoping, well, no, we are in 2026 to bring together the CFD members and other members of other resources across NIH to talk about ways that we can create interconnections and what are the ways to limit the barriers for other people who are outside of all of these resources who want to use the data within. So that's one activity. So if you're part of a resource, come reach out to us. We'd love to have you at that event. Another one, we have funding, travel funds to get you there. Another one is experimental validation. So basically, what we've created here has a lot of hopefully low-hanging fruit for folks who actually want to validate these findings. So we actually have funds to deploy this current year to experiments. If you find a hypothesis and you want to conduct an experiment to validate that within the Knowledge Center, we have funds to allow you to do that we can deploy. So that's another really cool component. And then the last one is for those computationally minded, we also want to engage you. So you heard about me talk about some of the secondary analyses and some of the tertiary analogies. We really want to engage computational users to take Common Fund data, both inside the Common Fund, any data within the Common Fund, and external resources to create new analyses. And we have, serial hackathons and data challenges set up across the year. You'll hear more about this, but what's cool about this is we have prizes. We're going to feature you. We really hope that you get inspired to actually generate some different ideas and analyses that we would then make accessible on the Knowledge Center and the CFD at large. And this will hopefully lead to new collaborations. So these are 3 activities that we hope to continue the momentum of the Common Fund in the coming year. So with that, I'll stop and thank the people in my lovely ecosystem that I get to work with that build what you just heard me talk about today. And with that, we can stop and take questions, or I can do another demo of any question that you guys might want to have me ask.
Just a reminder, feel free to ask questions either by raising your hand, using the chat, or using the question and answer. We're monitoring all 3.
And as Mackenzie said, these are recorded. So if you have any questions about them, you can see the recording, or you can email us afterwards as well. Julie.
Yeah, I guess I'll get us started. So this is more a question for the audience, actually. I see we have some experimental folks who are here with us on the call. We would love to know, how would you use a resource like this? What types of questions would you want to ask about your biology, your disease area of interest? I don't know, do you have some sample prompts that we might be able to test out and see how they work using this new tool?
I have it open and ready to go.
You can also answer in the chat if you are uncomfortable speaking.
All right, well, no one to hold everybody up. So if you have any questions, play with it. Tell me what you think. Tell us what you think. And tune in for what's our next webinar, Mackenzie, actually,
Yeah, so it's going to be rescheduled because it's currently for Thanksgiving, so it's going to be the week before. So that'll be there. And then we have it booked, but just give me a second for the actual crew.
No worries. All right. Any last questions or comments from people? Actually, one thing I wanted to mention, the training center is really good at training, as we've learned working with them. So what they've agreed to do is help us make this easier for you to use, because I can imagine I kind of threw a lot at you and described a lot of what's underlying this, which is not necessarily super, easy to unpack. At least for me, it's not. So I thought they're going to be our partners in really making this more useful. So hopefully there'll be a training video or whatever mechanism they think's best for you all to use to actually use this a little bit more easily within your work.
Just one thing, so it's going to be on the 21st, just a reminder to everyone, and it's going to be IDG. So we're really looking forward to that. And we have so many more exciting webinars coming out in the future. There is a question. There's a couple questions that are actually popping up now, which is really great that we haven't logged off yet. One question from Brendan Riley. Are you involved with the Hack Medicine event on November 8th? Should I show this to people I am mentoring there?
Yes, yes, yes. I'd love to. That'd be great. We're not involved. I've never heard of it actually. So, Brendan, if you'd like to actually email us afterwards, Mackenzie can pop either my email in the chat, and I'd love to actually hear about that. And if you are interested in mentioning this to people that you're mentoring there, that would be great. And we can give you any more information you'd like. We would appreciate that a lot.
There's one other question. Is there documentation of all the relationships modeled in the CFDE reveal?
Good question. Yes, and yes. Julie, is that correct? So for pigeon, that's documented, the method. Yes, I'm gonna let Julie answer this.
So I guess it depends on what you mean by all the relationships. So the what we have documented is the pigeon method that Noel described and how that works, where the gene sets are coming from, and that sort of information. If you're asking, let's say you give an LLM prompt, that is, what's shown here. So which pathways associated with exercise in the liver are, whatever that prompt is, and you want to know exactly what all the possible um, relationships are that this type of a prompt can answer, that is not something we have done sort of extensively.
Yeah. But if you go to, right, if you don't know if you saw me click it, right here, I went to the wrong one, shoot. Oh, there's reveal, let's go down here. If you go here, so this is view of the data where you can interact, but the research method here does give you some things. It gives you how the semantic search works. You also have pigeon, as you mentioned, how the embeddings are made, and then how we make like this is the pigeon documentation that Julie was referring to. So there is documentation on the core data and method that you have. But you're right, the enhanced workflow is something that we need to add.
And just to clarify, were those the types of relationships that you were asking about, or were you asking about something a little different?
Or sorry, types of relationships.
Okay, great. Thank you.
Cool. All right, Brendan, thank you. I'll send you a direct message actually. Any other questions?
Okay, well, if you think of any questions between now and our next webinar, please feel free to reach out to us. We are more than happy to schedule some one-on-one time as well to go over these features. And we'd be happy to, answer any questions, concerns, feedback, etc. So thank you everyone so much for joining us today. Reminder, our next webinar will be on the 21st, and we will see you soon. Thanks, everyone. Bye.